Okay, everybody, what we're going to do now is we're going to go into cellular respiration, um, which is actually a really important topic because it talks about how chemical energy is stored in food. Not only that, but it shows how you pull it out to run all of your biochemistry. And so I found this quote which I really liked. It says, "Food is basically solar energy that's stored in the form of complex biochemical substances." And I kind of like that. So we're going to see how the things that we take in from chemical energy, such as carbohydrates, fats, proteins, and some other things, um, end up allowing our body to produce ATP, which we've seen runs a lot of our biochemistry um, in the cell. So, just an overview. So, our bodies are going to somehow need to acquire glucose to make this chemical, to make ATP. And we know that plants produce it through photosynthesis. We're going to touch on photosynthesis later. And that all other organisms have to attain it through food. And so in our case, that is going to be plants, right? And that we can go ahead and store that if we have excess as glycogen or starch, again, if it's plants. And we're going to see that it takes ATP to make ATP. And so initially, we're going to invest 2 ATP to go into this process, and we're going to get out somewhere between 30 and 32 ATP molecules back. That's an amazing investment because the payoff is huge. The payoff is huge. So we have to ask, okay, what happens when glucose is oxidized? And to just go back and review that. So what does it mean to be oxidized or or to be reduced? So if we see here on the right hand side, we have any type of chemical. It can be a sugar, as we're talking about. It can be a protein. Um, and so here we have that particular chemical. And we see that in this case, when it loses electrons, right? When it loses electrons, um, we see that it becomes oxidized. Over here, we have chemical B. We see that if it gains electrons, then it becomes reduced. So, given that being the definition, right, we're going to see how glucose is oxidized, right? How is it oxidized? Where we're going to see this play out in our cells in all 50 to 70 trillion of them. Okay, and so when this happens out in the environment, let's say we take glucose and we burn it, right? So we know that we're going to see energy released, we're going to feel the heat, we're going to see the light. But that's a little bit different from what happens in the cells. The cells are going to use it differently to produce the energy that they need. So inside our cells, right, glucose is oxidized, and it's a long process. We're going to see that through four steps. Um, and what you basically see is you see glucose coming in, and then we're going to use the oxygen that we breathed in, carried by red blood cells, and that in the cells, in the 50 to 70 trillion cells of our body, these two are going to be used to then produce carbon dioxide, water, and most importantly for what we're doing today, ATP. That's our energy. There's another process also called fermentation where we can also process glucose and we can oxidize it. And that will also produce ATP, but in the absence of oxygen, we can't fully oxidize glucose. So you don't get um, as much ATP out of this if it's an anaerobic reaction. That means without oxygen, which is something that happens if you've ever felt your muscles start to burn, right? That's because the lactic acid is building up. The cells have run out of that oxygen, and now they're doing an anaerobic process. And that's what you start to feel. You start to feel that burn from the lactic acid. You are still getting ATP from the oxygen, but it's not nearly as efficient. And so glucose is central, right? We need this. So we're going to need glucose to go through this process of four major steps so that we can recover, as we said, 30 to 32 ATPs for every two ATPs that go in. So this is our energy source to generate ATP. And because if we remember, the sugars are basically carbon chains, this is also our carbon source. This is also our carbon source. So here are the four major steps, and we're going to see these. We're going to see glycolysis first. That's where we're going to break down the glucose that you brought in from your diet. That can be coming in from vegetables or from beverages, all sorts of sources. We're going to use that process to create pyruvate. We're going to see that. And then we need to process that. That's going to give us acetyl-CoA. Then we're going to use that in the citric acid cycle. That's part 3. And we're going to see that. You probably have heard of that. It's also known as the Krebs cycle. And then finally, we're going to look at the electron transport. And there's an oxidative phosphorylation step at the very end, which gives us our ATP. So if we look at it, it's going to look like this. Step 1, glycolysis, glucose comes in. We're going to convert that to pyruvate. We're going to take that into the mitochondria. We're going to see how we process that to make what we call acetyl-CoA. That will then go into the citric acid cycle, also known as the Krebs cycle. And we're going to see how after that, we're going to use some of our products to run the electron transport chain and give us back far more ATPs than we put in. And in each step, we can see here, we can list what goes in, and what comes out. So we're going to be doing that step by step as we go through this process. Here are some of the questions as we go through this process that we should keep asking ourselves, just so we don't lose sight of what's going on. So in each step, what is going in? What is going in? What is coming out? There's going to be a biochemical process. What is coming out? What happens to the energy that's released? That's an important question. Where does each step occur in the cell? And how's it regulated? So these are the questions. But there's something I want everyone to remember, and that is when we have these biochemical processes occur, all of this is driven by electrons wanting to be in their most stable state. And we've seen that happen with chemical reactions. We've seen enzymes come in and help that by lowering activation energy. But at each point, we see what's going on here on the right-hand side, where on the y-axis we see energy, on the x-axis we see this process that's going on, given any particular process, where at step 1, you have electrons with a certain amount of energy, right? And sometimes you have to push that uphill with some investment of energy, and then they'll drop down to where we see step 3. And now the electrons will be happier. They're going to be more stable there. So all of these steps that we're going to see is basically these electrons trying to get to a more stable state. So we're going to see some catabolic pathways. Catabolic pathways, as we can see on the bottom left, That's when we break things down and we can get energy out of that. Remember, energy is stored in the chemical bonds. So as you're breaking things down and releasing those chemical bonds, you're using that energy for something. We're also going to see many anabolic pathways. This is where you actually synthesize things, you build things up. Okay. And so we can see here in the middle of the bottom screen, but we're going to use energy to build things. You may have heard that word before, anabolic. Anabolic steroids have been in the news for decades. And this is because many people have used them to encourage their cells, right, to synthesize more protein. And that's the bulk that you see. That is where you see the increase in the muscle, because this anabolic steroid is inducing these cells to build up, to synthesize more protein. And you can see that in the muscle mass. So we may have heard anabolic, so you can remember body building, building. Anabolic pathways build something, whereas the catabolic pathways tear stuff down. Okay, you can actually use many different types of molecules to get energy, to produce ATP. So the ones that we're familiar with are carbohydrates, right? Those are our sugars. But in the absence of sugars, your your body can still run itself if it has supply of fat. What's going to happen, as we see on the diagram, it'll take fats and through other biochemical processes, right, it's going to catabolize those. It's going to break those down into things like glycerol, fatty acids, and those can be used also in the Krebs cycle to produce the ATP. As a last resort, the body can start to consume itself within its cells. It can start to use proteins. It'll break them down into amino acids. It'll get rid of the functional groups, and everything else will go into the citric acid cycle. And so, by any means necessary, your body is going to find a way to produce ATP so that it can run all of its biochemistry. And so when we look at this process, which we can see in the middle of the screen from left to right, we're going to start with glucose in red, and we're going to see these steps. If we remember, we're going to make pyruvate, then we're going to make acetyl-CoA, and that's going to go into the citric acid cycle. That's what we're looking at today. Remember, the glucose comes in from food. Yes, it can be fats, it can be proteins if sugar isn't available. But when we look at this, we see that there's a lot of things that this pathway touches on, right? So catabolic pathways, we see the breakdown of sugar, we see the breakdown of fats, the breakdown of proteins. But in the anabolic pathways, we see that glucose being in excess, where we then build glycogen, and that's what we store. Plants use it and they build starch, and that's where they store it. We can also see that in this pathway, we actually get certain substances, what we'll call substrates, to synthesize nucleotides. Those are the things that we use to build our DNA and our RNA. Later on, we see that we can build up phospholipids. We've seen that. That's super important for our membrane. We can build up fats. And if we keep going to the right, looking at the citric acid cycle or the Krebs cycle, some of the things that we build there, we use to build amino acids. Okay, the ones that we do build. Remember, plants can build all 20. We can't build all 20. But when we do build them, we can see that this particular pathway is involved in all of that. And this is kind of a bird's eye view to see, okay, that middle pathway, which is a more bold, darkened gray pathway, it is touching on sugars, lipids, nucleic acids, and amino acids. Okay, remember, we said there's four. There's four steps. So we're going to get to the first step, which is glycolysis. You can remember that because of glucose, right? That glucose came in from that beverage or from um, that plant, that vegetable. So that's going to be the first part. And there's 10 chemical reactions actually used to accomplish this. In this particular step, this is where we put in the investment of 2 ATP. And in these 10 steps, what we're going to get out is we're going to get those 2 ATPs back. We're going to get some NADH. Okay, that is going to be an electron carrier for us, which is going to be really important later on. And we're going to produce 2 pyruvates. Remember, those pyruvates are what keep moving down this pathway to eventually give us some 30 or so ATPs. So those pyruvates are really, really important. And remember, as we're doing this, we're going to see this many times over, the phosphorylation, right? So in this case, we have ADP, we have an enzyme that's going to take a phosphate group and link it to the ADP to give us the ATP. So we're not going to show all the enzymes, but they're there. We're gonna represent them as numbers, let's say, but we're not going to see all the enzymes up front. We're going to see maybe a few of them. Let's take a look. Okay, this is our first step, right? There's 10 chemical reactions here. So where's this going on? Well, this is going on in the cytosol, right? So what's going in? Well, we see we're investing those 2 ATPs. Here's our glucose, and we pluck off one of those phosphates, and you see it there. Now, there's the phosphorylation. We have a processing step here where this sugar, which has 5 carbons, right? 1, 2, 3, 4, 5, and then that's an oxygen, right? This gets modified down to 4 carbons. Now, at each step, we see a green number. That represents the enzyme. Remember the Olympic relays. This enzyme, represented by a green 1, will take glucose and modify it and then create the next substrate, right? And pass that on as a baton to enzyme number 2. Enzyme number 2 will take that, it'll modify it, and then pass it on to enzyme number 3. Enzyme number 3 will take in another ATP and cause another phosphorylation. Okay, so we see the relays, we see the relay race. In this case, the step that's important is we're actually then going to cut that, and what we get is we get 2 species that are 3 carbons long. So remember, we reduced our carbon number. So there's 1, 2, 3, 4 in the ring. Here's our 5th carbon up here. Here's our 6th carbon up here. So we're going to get 2 of these 3 carbon chains. And so now we're going to start to produce things. In other words, what comes out? NAD is one of our electron carriers. Remember, electrons want to go somewhere where they are more stable. And so NAD is going to be taking those. And so how are they going to be taking them? Well, in the form of hydrogens. Those hydrogens have electrons. So the NAD is going to be taking those electrons here. Again, we see more enzymes encoded in green. Here we're taking phosphates. That I means inorganic phosphate, which means it's not attached to any carbon or anything, it's floating around. And so we've got more enzymes, and they're phosphorylating. Remember, we talked about that. They're phosphorylating these chemical structures here. So this is 1,3-bisphosphate glycerate. So these chemical structures. Here we're getting phosphorylated here. And now what we're going to do is we're going to take 2 ADPs and transfer those phosphates. And look, we got back the 2 ATP that we put in. We've got some more enzymes. They're going to do some modifications. So here we lost a phosphate group. but it was two of them because remember from the last slide, we have two of these species that are 3 carbon chains long. That's where these twos are coming from here. So in this case, we have 2 of these species. We get some modification from an enzyme. We have 2 of these, and so forth. We're producing some water. So remember, that's a condensation reaction. Until finally we get to the end, we see that we have 2 of these species with an extra phosphate. So we're going to take those off and give them to ADPs. Now we've produced 2 more ATPs. Now we're up by 2. We invested 2, we got those out, and now we've got 2 more. So we're already up, but the investment's going to pay off a lot more than that. And finally, we get to pyruvate. And we kind of stop here because remember, this is going on in the cytoplasm. Pyruvate is going to enter a different part of the cell where we're going to continue this. But this whole thing has to be regulated. So we have 10 chemical steps. We have 10 chemical steps. And in this third step here, we can slow this thing down. How do we slow this thing down? Well, that third enzyme is this enzyme here. And here's the thing, if the process is going too fast and the ATP is building up, then excess ATP will bind enzyme 3. Here's the name for enzyme 3, phosphofructokinase. So the important thing here is we need to slow things down. Think of it as a decelerator, a brake in the car. We are taking up all this sugar. If we're producing more ATP than we need right now, then that excess ATP binds enzyme number 3. Why is that important? Because we remember when you bind ATPs, you change shape and you start to turn off. And so if there's too much ATP, that excess binds enzyme number 3, which we're staring at on the screen right now. It changes its shape and it's not functioning. So now what we've done is we've slowed down the process. When ATP comes down in concentration, these ATPs will let go. This thing turns on, and now we can start up the process again. So that's the part of glycolysis. Again, we have a product. That product is called pyruvate. These are the 10 steps, and we call that glycolysis before we move on to the next step, because the next step goes somewhere else. That first step with 10 chemical reactions was in the cytoplasm. But now we go into the mitochondria, which we see here. The mitochondria is going to take up pyruvate, which we just made from the glucose that came in. And when it takes it up, it's going to process it using more enzymes, right? So now we have 2 pyruvates. We put in 1 glucose, we got 2 pyruvates out. Okay, so then what's going to happen inside the mitochondria? It's going to take up the pyruvate. The enzymes exist in these ruffles that we see here called cristae. Those enzymes are going to see the pyruvate. And again, the relay race, the Olympic relay race, continues. So what goes in? Pyruvate that just came into the mitochondria. Right? We're going to take off that CO2. So there's the CO2 production there. So we're making CO2. And what we're going to do is we're going to link a helper. This is coenzyme A. Coenzyme A is going to help us with the next process. And if you see there's a sulfur there, that sulfur is holding a hydrogen. We need to get rid of that because we want to link that sulfur to what's left of the pyruvate, and there it is. The pyruvate came in, we take off the CO2 and we put the coenzyme A on it. That hydrogen is extra, So we put that on NAD. That's one of our electron carriers. The hydrogens carry electrons. So here we go. Now we have NADH, and these are going to be really important later on. You wouldn't think so just because we haven't been talking about them very much, but they're going to be really, really important. And so we're building up a lot of NADH, and we're going to see that from these coenzyme A's. Okay, so that that was pretty simple. That's all there is in the second step, taking pyruvate to acetyl-CoA. And this we also regulate in the same way. So here's pyruvate dehydrogenase. That was the enzyme that was doing the work. And as you're taking pyruvate and the coenzyme A and the NAD, which is our electron carriers, if we're making more ATP in excess, once again, this enzyme can be phosphorylated. There's the phosphate group there. What does that mean? It means it just changed shape and it's not functioning. That's why it says inactive. If the ATP comes down in concentration, now we're ready to go again. The phosphate comes off. The enzyme is back to its original shape. It's ready to go. It's receiving pyruvate. Remember, this came in from the cytoplasm, and now it keeps going with regard to making acetyl-CoA. Acetyl-CoA is important because that's going to be the beginning of our next step. Okay, the famous citric acid cycle, also known as the Krebs cycle. So we've been here in the cytoplasm, making pyruvate. We came in to the mitochondria. We converted pyruvate to acetyl-CoA, and now we enter the Krebs cycle. And so the Krebs cycle is a really complex cycle where you're going to have a lot of different enzymes. So remember, where are we? Within your 50 to 70 trillion cells, after you ate that burger and had that juice, right? Remember how we digest all of that. But now this has gotten into the cell. The sugar's gotten into the cell. Now we have to produce that ATP. We did glycolysis. Then we moved into the mitochondria. We converted pyruvate to acetyl-CoA. We're going to remain in the mitochondria now, but we're going to go into our next cycle. This is the citric acid cycle we call the Krebs cycle. So look at all of this. I'm not going to make you memorize any of this. However, some of my colleagues in the Department of Chemistry, if you ever take biochemistry as a course, will make you memorize all of this. And they'll probably make you draw it as well. I remember from my undergrad days, before smartphones, we'd have to know this stuff. And every time I see this particular product here, brings back memories. I remember we used to cheer just because you'd use anything to memorize all this stuff. 2,4,6,8 alpha ketoglutarate. And that would get us past that step. And so we don't have to memorize any of this, but we do have to understand what's going on. In the mitochondria, pyruvate was taken to acetyl-CoA, and now that's going to drive the citric acid cycle. Notice again, we have a lot of enzymes. They are in green. These are the relay runners. Acetyl-CoA comes in. Here's our first enzyme. It grabs that baton, and from it, it'll convert it and it'll make citrate. It's taking two batons, the acetyl-CoA, right? And the oxaloacetate, we'll get to that later. That puts them together, and now we have citrate. And now here comes the next Olympic runner. This is an enzyme. Takes this chemical, takes this baton, converts it to the next. Next runner takes this chemical as its baton. It's going to get rid of a hydrogen. Look, there's another NADH. Converts it to alpha ketoglutarate. Another enzyme comes in, pulls off another hydrogen, Now we've got another NADH. So we've got all these electrons moving onto the NADs where they're happier because their energy level is lower. And we keep going. We're making some ATPs or GTPs. We haven't talked about those so much yet. They run a lot of biochemistry too. Now we keep going. Here we've got our sixth runner. It's going to take one of those hydrogens and add it to a different electron chain participant. So in this case, it's FADH2 taking those hydrogens, which have those electrons, right? In this case, taking two. So we're loading up on electron acceptors, right? We've got lots of NADHs now, lots of FADH2s. They are taking the electrons, making the electrons happier because they have a lower state of energy. But we're going to use all of these. Notice H's are building up everywhere. NADHs, FADH2. So we're going to do something with all of these hydrogens. These are protons, right, with electrons around them. We're going to keep going. Here's another NADH. So again, we've just got lots of these chemicals accepting electrons and protons. That's what the hydrogens are, right? And so we're making a lot of NADH, a lot of FADH2s. We're making some ATP, some GTP, not a lot, but we're recovering more. So our investment gets even better. So as I said, I don't make you memorize all this, but just remember acetyl-CoA goes in and we start to build up NADH and FADH2. Those hydrogens carrying electrons are going to be super important for later on. And that's our third step. That's our third step. Here are the steps. For you to go back and make sense of any of it, you don't need to know all the details. Not until you take Chem 3270. Okay, but what we've done here is we've brought in acetyl-CoA. Remember, that was made in the mitochondria when pyruvate came in from the cytoplasm. And again, just to repeat it, we have more feedback inhibition here. So we're regulating this here as well. So every step is regulated. Every step is regulated to make sure that the process doesn't speed up too fast. If we're not needing to convert as much sugar because there's more than enough, we slow things down and we keep the pace that the cells are used to keeping. Okay, and so again, there are enzymes that will interact with an ATP, change shape, and stall the process. So the theme keeps going. Okay, let's pull back for a second, because that's a lot of information. You went out to lunch. You had your beverage, your meal, maybe a dessert. Here comes the glucose. Glucose was taken into a process that we call glycolysis. Remember, that had 10 steps in it. From that, we got 2 pyruvates out of that. We call that the endoglycolysis. Those 2 pyruvates went into the mitochondria. We had a short process where that pyruvate was made into acetyl-CoA. So now we've got 2 of those. And we made some NADH, electrons moving on those hydrogens to lower states where they're happy. Okay, the acetyl-CoA goes through that very complicated citric acid cycle, also known as the Krebs cycle. We get 2 more ATPs. So again, we're getting our investment back. But look at this. We built up 6 NADHs and 2 FADH2s. Now notice each of those has 2 hydrogens, right? So we're building all of this up. They're taking these electrons. They're taking these electrons. And remember, when you take electrons, right, you're being reduced. Everything else, which was the glucose and everything else, is being oxidized. Okay, there must be a reason why we're building all of this up, right? Well, we're going to get to. The question is, so what is all this for? Where is it going on? Again, we're on our way to making a lot more ATP. Right now, we happen to be in the mitochondria. Having gone through the outer membrane and the inner membrane, we're in the matrix. You see it labeled here. So again, these electron carriers, the NADH, the FADH2, right? They're going to take those electrons, and what they're going to do is they're going to introduce them into the electron transport chain, which we've all heard about. Okay, remember what I said? Electrons are looking to find the most stable state they can. The most stable state they can find, they're going to go that way. And so when we look at it and we look at the enzyme stability, here we start with glucose. We had to invest 2 ATPs, get over some of that activation energy. And after that, electrons, they just start losing all this energy. They are powering a lot of this chemistry because, again, electrons form bonds. So all of these enzymes are converting chemicals. And what are they doing? They're just pushing electrons. That is all they're doing. All of these enzymes that we've seen are just pushing electrons around until they're happier and happier and happier and happier. And you can see the energy that's being released. This is what's powering this. They want to go to a lower state, lower state, lower state. And this is what's allowing all of this chemistry to happen. The electrons are happy to go, happy to go. And along the way, we see we're building up NADHs and FADH2s, specifically because the electrons are happy, but we're going to use these. We're going to use these. And so, okay, we went through the first 3 or 4 stages. We did glycolysis. That was in the cytoplasm. Then we processed pyruvate. That was in the mitochondria. We got acetyl-CoA out of it. We remain in the mitochondria. And now as we see, we are building up tons of NADHs, tons of FADH2s. So the question is, what happens to all the energy that's being released as they're being transferred? Well, again, that energy is powering the formation of the next bonds. As the electrons drop down energy levels. So this is what's powering. Okay, how is this used to make ATP? Well, we saw the first 3 steps. Now we're going into the 4th, and we're going to see exactly what it is that's happening. We're going to look at the electron transport chain. You probably all heard of it. You probably had to study it back in high school. So now we have all of these chemicals that have taken the electrons. Specifically, I'm talking about the NADHs, the FADH2s, and we're going to see how we're going to oxidize those, which means other proteins are going to be reduced. Okay, and so we're going to see one important protein. Actually, we're going to see 2. We're going to see 2. One is coenzyme Q, and we're going to see the other in a moment. That's really important, which is cytochrome C. It's important for many reasons, but right now we're just looking at its role as a carrier in the electron transport chain. So We've got plenty of electrons and protons coming in from those hydrogens. So we just used up 2 ATPs to build up tons of these. And so what do you think is going to happen next? The same thing. These electrons are going to be looking for more stable states. When we look at free energy, remember how much we dropped in making all these NADHs and FADH2s? Well, we're not done. So we're going to be dropping in more free energy. Again, electrons wanting to go to a more stable state, that's what drives all of this. So here we have coenzyme Q. We have another protein called cytochrome C. And these two are the ones that are going to be transferring electrons to the different complexes that make up the electron transport chain. There's going to be 4 major complexes. So here's complex 1. We see coenzyme Q transferring electrons. Those electrons are going to keep going down the energy gradient, transferring to more complexes, all the way down until they've lost just about all of their potential energy. The electron transport chain, again, it has many components. You're going to have a blast in biochemistry memorizing all of this, but not here, but not here. Okay, so let's take a look at what's going on. Remember, we built up all of those NADHs, all of those FADH2s. That was the result of the glucose coming in. Yes, we had made some more ATP, which was great. But really, the most product that we were producing were these two. Okay, so then what was that all for? Well, we see them here. Now, where are we right now? We see that we're still in the mitochondria. There's the matrix inside. Remember, the mitochondria has 2 membranes. Here's the inner membrane. Here's the intermembrane space. And you don't see it, but above that, you would have the outer membrane, on the other side of which would be the cytoplasm. And so here we are inside the matrix. This is that deep part of the mitochondria. And here are NADHs and our FADH2s. And what we see is these two species are now losing all of these electrons, which are being carried by these protons. So remember, 1 proton gives you hydrogen. 1 proton gives you hydrogen. So as they're losing all of these hydrogens, those hydrogens are carrying the electrons in those orbitals. And they're starting to build up. You see here, they're starting to build up in the intermembrane space, in between the inner membrane and the outer membrane. In between. So they're starting to build up. We see the processing of these species by complex 1 and complex 2. We're going to continue this on. So now what we see here is we see Q, and then we're going to see cytochrome C. And what's happening is they are taking the electrons. Remember, they are now our main electron donors. They're taking the electrons off. So what happens to all of these hydrogens? When you're taking the electrons off, the hydrogens, because they don't have the negative electron, they're no longer neutral. That's where you get all these H pluses. Well, where do the electrons go? Well, that's why we have coenzyme Q and cytochrome C. They're taking them all. Right? And so now we have positively charged hydrogens. They're protons, but they don't have the electron in the orbital anymore. It got taken by coenzyme Q and cytochrome C. And so here we're building up this gradient inside the um, inside the mitochondria. And so what are we doing all this for? So again, glucose came in, we built up tons of this. Now it seems like we're stripping a lot of electrons and we're taking a lot of hydrogens. If we remember weeks ago, we formed gradients inside and outside of the cell. You will remember sodium, you will remember chloride, you will remember potassium. And when you have a high concentration of one of those, it wants to diffuse into areas where there's low concentration. And that helped us power a lot of things, like the glucose transporters, other stuff that we talked about as well. So now what we've done is we have taken the hydrogens from all the NADHs, all the FADH2s, put them in the intermembrane space between the inner and the outer membrane. And now we've created a gradient. And what happens when you create that gradient? Things want to force themselves into the areas of lesser concentration. And that's exactly what happens here. It's pretty amazing because it's a motor. It's a motor. This is ATP synthase. So you can think of synthesis, right? When you make stuff. So in this case, what we have is we have ATP synthase being a motor. It's driven by the hydrogens. It's driven by the hydrogens. And you can see here it's got two units. We've got the F0 unit and the F1 unit. You can see the F0 here. And the hydrogens power this motor so that what it does is that it ends up attaching phosphates to all the ADPs that are in the mitochondria. Let's take a look. This is the step that garners us 30 to 32 ATPs. So we see it here. We see the hydrogens spinning the motor. If we look inside, here we see the ADPs coming in. The ATPs are going to be phosphorylated. In other words, you have an adenosine with two phosphates. This is the thing in the F1 space here, in the F1 subunit, where it's going to attach an additional phosphate. There it is there, right? And it's going to attach it, and you have a triphosphate now. That glucose that came in when you had that donut, when you had that soda, what it ended up doing is it ended up being oxidized reducing all the NADs and all the FADs. The purpose of that was to create a gradient that drives a motor. And what that motor does is it connects phosphates to ADPs. It's almost like it's electricity, right? I don't necessarily like that analogy so much, but in a way, right? When you plug something into the wall, the electrons just start flowing. So the glucose, the sugar, built up the, the gradient in all of those steps that we saw. It built up that gradient. And the reason that's important is because that gradient is going to cause a current of hydrogens to power this motor. I think we've all seen either recreations or animations of the 1600s and 1700s where they had those massive wheels in rivers, right? And the river would push the buckets on the wheels and it would cause it to turn. You can see it almost the same way here, where the hydrogens are causing this thing to turn. The change in shape facilitates the pushing of electrons, and you get phosphates being attached to ADPs. So that's what the sugar was for. And again, if you don't have sugar, your cells will use fats. If you don't have fats, they'll start to cannibalize themselves and use their proteins, breaking them down and using the amino acids to drive all of this. And so over here on the left, again, we see the hydrogens building up. Here we see the ATP synthase that is turning like a motor and linking all of those inorganic phosphates. These are the phosphates that are just floating around. That links these inorganic phosphates to ADPs, charging them again with a third phosphate. And now those phosphates can be used to change the shape of proteins, activating, deactivating, and so forth. So if we put it all together, how does your food store energy? This is solar energy, right? How does it store that? Well, it stores it in starch. Those are just chains of glucose molecules, right? Again, we can take up glucose in just a monomer form if we're drinking a soda. Here comes all of that glucose. And we spend time both in glycolysis and in pyruvate processing and in the citric acid cycle, not just building up more ATP, we get some, but we spend that glucose building up hydrogen carriers. Remember, these hydrogens carry electrons, so these are electron carriers as well. We keep building these up more and more and more. So that later on, we have coenzyme Q and cytochrome C helping separate these two, the hydrogen from its electrons. That hydrogen buildup inside, right, is going to try to push its way through back into the matrix because everything likes to diffuse, and that spins ATP synthase. That constant changing of shape causes the electron pushing so that it binds phosphates floating around onto ADPs, creating ATPs. Your book says about 29. Most sources say between 30 and 38. Usually they say between 30 and 32. I'm not sure why your book says 29. Most sources say somewhere between 30 and 32 is what we get back after investing too. How does your food store energy? It produces glucose, which your cells use to build up a hydrogen gradient, which, like an electrical current, runs a motor that attaches phosphates. And remember, when we look on the back of a label, right? When we look at the label on the back of vitamins, what do you see? Phosphate. Phosphate also comes in your diet. So this is something that is throughout your body, and it's taking those phosphates and latching them on to the ADP to create ATP, which is going to be for shape changing. Now, I know that was a lot of detail. If you heard me repeat things over and over, you know it's important. I'm not going to ask you to draw any of the chemical structures, but you need to know at each step, remember there's 4 major steps. We need to know at each step what is going in, what is it that we're using at that particular moment in the process. We need to know what we're getting out. Just that we're getting out from those 10 reactions in glycolysis. Where is this happening? Is this step in the mitochondria? Is this step in the cytoplasm? Is it regulated? How is it regulated? You see the repeating pattern. ATP builds up and it starts to bind stuff. It starts to bind the enzymes themselves, and they change shape and then they don't work. ATP goes down, and then those phosphates start to come off. The enzymes go back to their original shapes, and they start to produce more ATP. So what is going in? What is coming out? Where is each step occurring? How is it regulated? You should be able to follow your French fry all the way down to the ATPs that are helping run all the molecular biology in your body, all of the biochemistry in your body. So that's how your food becomes energy, because that's what's going to turn things on and off, the ATP, particularly the phosphate.